Welcome to our second podcast on blood. We're going to continue looking at blood cells, and we're going to pick it up with the agranulocytes. We'll touch on the platelets, and then we'll move into blood cell development or hematopoiesis. So our agranular leukocytes we'll describe are the lymphocytes and the monocytes. We're also going to describe the components of the platelets and their activation as far as forming a platelet plug. And then we're going to identify and classify cells that we will see represented in bone marrow that will represent the different stages of hemo or hematopoiesis. So our first agranulocyte we're going to look at are the monocytes. Now these are going to be the largest of the circulating cells we'll see in a peripheral blood smear, maybe 2 times or 3 times larger in diameter than we'll see with a red blood cell. You're going to see maybe anywhere from 5 to 10% of the white blood cell population as monocytes, so this will not be uncommon when you see these cells in peripheral blood. Now, the nucleus is going to be somewhat kidney-shaped, we say indented in nature, and the cytoplasm is going to look bubbly. And this bubbly, soap bubbly look appearance to the cytoplasm because of all the phagolysosomes and the phagosomes and the endosomes that are present in the cytoplasm from material that's been phagocytosed. So you've got the indented nucleus and the bubbly vesicular cytoplasm are indicative of these fairly large cells we'll see in the circulation. Now, macrophages, monocytes in the circulation, that is, they will be there for a few days, but they will move out into the tissues where there'll be macrophages, and they will stay possibly several they will stay in the tissues and possibly survive there for several months. So here we see our diagram, the indented nucleus of a macrophage. There on the right, we can see the relative sizes, very, very large. And then as we look in the cytoplasm, often we will see that soap bubbly appearance. Well, specifically, we'll really see that when we look at a transmission electron micrograph. In this, it's not quite as evident. It almost looks like mottled in the cytoplasm, but here we can easily see the indented nucleus. And if you see red blood cells, you know you're in a peripheral blood smear, and that is a monocyte and not a macrophage. So macrophages, always say they're the vacuum cleaners of the body. They're going to phagocytose cellular debris. They're going to produce cytokines, lymphokines, because that's going to recruit lymphocytes to the area. This is going to recruit other immune system cells. But the biggest thing that I want us to remember is these are APCs, antigen-presenting cells. And they're going to present these antigens to lymphocytes. And the way this happens is they endocytose this material. They are going to display those epitopes on their cell surface. And they're going to do so through our HLA class 2, or what we learned in physiology, our major histocompatibility complex, MHC2 markers, and therefore display those antigens to the lymphocytes. Here when you see an example of how that's going to happen, there's our MHC protein on the left. You can see we bring in the antigen, the debris, that's coupled with our MHC2 protein, then that's displayed on the cell surface to present to our lymphocytes. Now, lymphocytes, the last type of agranulocyte, are going to be approximately the same size as red blood cells, so they're fairly small, but they're abundant. 20 to 25% of our white blood cell population will be lymphocytes. These may survive a few months. They can survive for many years. Some lymphocytes may be around your entire life. So these are the longest lived of our white blood cells. Very easy to identify. They're mostly nucleus. You may see a very thin rim of cytoplasm around the edge, almost like a crescent shape, and that's going to be characteristic of what we see with lymphocytes. Here we see our illustration. Again, predominant nucleus. That's going to be the biggest thing that you're going to see. And in our peripheral blood smear, once again, you can see mostly nucleus, sort of a crescent of a rim of cytoplasm here. And the size is maybe a little bit larger than, but about the same size of a red blood cell. That's going to be our lymphocyte. And this nucleus, by and large, because of all this heterochromatin, it's going to be really, really dark in its staining pattern. Now with our lymphocytes, we realize from physiology, we've got the B lymphocytes that are going to give rise to plasma cells that produce antibodies. This is part of our humoral immunity. But then we also have our T lymphocytes that function with cell-mediated immunity. Now, as we look at any population of lymphocytes, say in a lymph node or in the spleen, predominantly we're going to have the T's, then we're going to have the B lymphocytes, and then we're going to have 5% of those are going to be somewhat of our stem cells, natural killers. We call these just the null cells. So B cells are produced and mature in the bone marrow. T cells become immunocompetent in the thymus. These are the organs where we get these names from. And really the B for B cells comes from the bursa in birds. It just so happens in mammals, we also do this in an organ that starts with the letter B. Now, once these T cells become immunocompetent, and once they are challenged with an antigen, then they begin to clone themselves. And as they do, we have two different cell types. We have the effector cells, which are either going to be the plasma cells to produce antibodies, or the helper Ts, the cytotoxic Ts, or we're going to make memory cells, memory B cells or memory T cells. And these cells are going to lay in reserve. You may think of them as being dormant until they encounter that particular antigen again, at which case we will mount a secondary immune response that happens very quickly. It's much more robust than our primary immune response. And generating those memory cells is the key for why we have our vaccinations against various diseases. So as I mentioned, B cells become plasma cells and they're the antibody producers. T cells, once they're challenged, will clone themselves and either become cytotoxic T cells, and they're going to destroy pathogens, or they're going to destroy virally infected cells. Whereas the helper Ts are going to initiate, or if they are regulatory Ts, in this case, they're going to suppress a T cell mediated response. And this is going to be done quite often by the production of lymphokines that are secreted and allow the immune system to either ramp itself up or begin to tune itself down. Here in our illustration, we see an immune response starting with a macrophage, and we can see the pathogen coming in. We can see the material being displayed on that MHC class II marker. That's going to encounter a helper T cell. And in this case, this helper T cell is going to recognize this MHC. Here we see a cytotoxic T cell recognizing that same combination. And in this case, the helper T is going to produce cytokines that are going to stimulate the growth and the cloning of our cytotoxic T. As this begins to clone, we have our cytotoxic T effector cell that's going to go back and find this infected cell, secreting its components of superoxides, the hydrogen peroxides, other materials that are going to lead this cell into an apoptotic cell death, that then this material can be removed by macrophages and other phagocytic cells. With a B cell response, here again we have a similar situation. We have a pathogen being engulfed by a macrophage, being recognized by a helper T cell, that helper T cell secreting its lymphokines and causing a B cell or a memory B cell to begin its cloning process and producing antibodies, these plasma cells, that recognize this particular epitope on this pathogen. You can see in that cloning process we also are going to generate our memory cells. Now that happens for T cells as well. They didn't have that in the previous slide, but understand that happens in T cells, and we have memory Ts as well as memory B cells. Here we have an illustration. We have a virally infected cell, and some of those viral particles are displayed on their surface with MHCs. And in this case, these markers are going to be recognized by cytotoxic T cells. And as they bind, again, they begin to produce what are collectively referred to as cytolytic enzymes. Again, the superoxide ion is one that does that as well. That leads then to an apoptotic or programmed cell death for the infected cell. And now we have phagocytic cells come in and remove that from the environment. So that's just an overall little animation of how that works. Now, we mentioned the null cells. These are either stem cells that can give rise to those other types of T cells that we have present in our circulation, or we have the NK cells, these natural killer cells. And these can also have cell-mediated assistance. They can basically find these foreign and virally infected cells, and they can, without the assistance of any other T cell population, launch a cell-mediated attack and actually destroy these cells on contact. And here we have a scanning electron micrograph that just shows an NK cell extending out processes, contacting a tumor cell and causing that tumor cell to undergo apoptosis. Now, the last part of our formed elements are the platelets. These are not cells, and so we say formed element. What we're going to do is we're going to start off with a large cell in our bone marrow, and we'll define that cell in a minute. And little pieces of that cell are going to pinch off. So you're going to have these little membrane-bound packages that we call platelets floating around in our peripheral blood. They're only about 2 to 4 microns, so a quarter to half the diameter of a red blood cell. Now, once again, we're going to have this sort of different regions, an inner region and an outer region. And in this one, the lighter region is going to be on the periphery, we call the halomere. And we're going to have a dark core, and that's called the granulomere. And these are going to contain a lot of materials that we're going to use in the formation of a platelet plug. Now, we may have upwards of 500,000 platelets per unit of blood, down to 250,000. And these are not going to be very long-lived, as you can imagine, these little packets. Somewhere in the neighborhood of 12 to 14 days will be the lifespan of an average platelet. In our peripheral blood smear, we can easily detect the red blood cells and what seems to be garbage or precipitated stain on the slide. The little bluish dots, those are going to be our platelets. And if we look at them at higher magnification, we can see that they may have these processes that are very sticky. It's very difficult to manipulate platelets outside of the body, and for this picture to occur, these platelets have been activated, and they have these processes. They're very sticky. They're aggregating and clumping together, and this is somewhat of an example of how a platelet plug may actually begin. Now, although platelets lack a nucleus, you can see we do have some mitochondria, we have cell surface receptors, and we have cytoskeletal components. So we're going to be looking at fibronectin, von Willebrand's factor binding sites. Those are integrin molecules. You see GP2B3A. That is a form of an integrin. Here we have other integrins for von Willebrand's factor. Here we have an integrin for collagen. And so these are going to bind those components of the extracellular matrix underlying endothelial cells. That's what these receptors will encounter if there's a vascular injury. And in many instances, binding of these receptors will trigger a second messenger response that's going to lead to the rapid degranulation and release of materials from these platelets, resulting in a platelet plug formation. Now we want to talk about hematopoiesis for a bit. Hematopoiesis, this is going to be the formation of new red and white blood cells. Erythropoiesis for red, leukopoiesis for white. But hematopoiesis, or hemopoiesis, some may say, is simply generic blood cell development. Now, in the red marrow, this is typically what we're going to see in newborns, and we're going to be producing a lot of red blood cells. But as we age, our marrow increasingly becomes filled with adipose tissue, and specifically unilocular adipose tissue. And at that point, we begin to call it yellow marrow. But we still have cell development, hematopoiesis going on, whether we're having red or yellow marrow. Now, as blood vessels enter and leave the marrow compartment, we refer to this as the vascular compartment because we have veins, arteries, and sinusoids. This is the vascular compartment, and vessels will drain these new cells that are moving into the circulation from the vascular compartment of the marrow. So between the trabeculae of the bone and this really spongy bone area, we're going to have hematopoietic compartments. That's within this vascular compartment. And it's filled with cells that have just so packed tightly together that we call them hematopoietic cords or hematopoietic islands. And this is really going to almost give this hollow out space its structure, just how the cells are packed into the spongy bone cavities themselves. Now, as we look at hematopoiesis, we're going to be looking at forming red blood cells because we have to form them very early in development. 2 weeks after fertilization, maybe up to 6 weeks, we're first going to be producing our red blood cells in what is referred to as the mesoblastic stage. And in this case, there is a layer of tissue. We have 3 layers that we're composed of, the endoderm, mesoderm, and the ectoderm, the mesoderm being the middle. And so this yolk sac mesoderm is going to begin to form what are called blood islands. And from this, you're going to produce blood vessels, but also blood cells from this mesoblastic phase in the yolk sac, really outside of the embryo itself. Now, from 6 weeks to birth, we enter into the hepatic phase, where we're going to be using the liver to produce as the location where we produce our blood cells. Now, early on, these cells are going to be nucleated, our red blood cells. And they're going to be formed first in the liver, but then we're going to start forming the white blood cells a little bit later, about 8 weeks after fertilization, during this hepatic stage. Now, also, the spleen is going to begin to take up its function and produce cells from the same 8 week to birth phase. And there's going to be overlap. They may not be doing it at exactly the same time. They may come on in staggered steps, but they both will play a role until we see about 24 weeks. This is the myeloid phase. This is when the bone marrow takes over, about 28 weeks. And so from 24, 28, 32 until 40 weeks, that's when typically delivery occurs in parturition. From birth on throughout your adult life, the myeloid phase is largely what does the production, the hematopoiesis in your body. Now, the liver and spleen may begin to do this later under certain conditions if you lose the bone marrow, but for all intents and purposes, after birth, the bone marrow is the location of hematopoiesis in the human body. So the bone marrow is going to be producing cells at a tremendous rate, 2.5 million red blood cells every second. So 10 to the 11th blood cells are produced in your bone marrow each and every day. And red cells, remember, are recycled. They have about a 120-day lifespan. They're broken down in the spleen or the liver if the spleen has been damaged. And a lot of this regulation of growth is going to be on the hormonal control that we have, either erythropoietin, leukopoietin, that are secreted by the body. Now, as we look at the bone marrow, we're going to have a lot of cells that we refer to as stem cells, but we're going to categorize them based on their potential to become a number of different cell types. And so we have these PHSC type of stem cells that we find, and these are going to be pluripotential hematopoietic stem cells. And these PHSCs can give rise to all blood cell types. So pluripotential means that it has a number of cells that it can give rise to and become. And when we look in the bone marrow, only 0.1% of nucleated cells there are going to be these pluripotential stem cells. From these, we are going to the next step then, after when the PHSCs divide, they're going to give rise to multipotential hematopoietic stem cells. And this is where we see a split. So we're going to give rise now to colony-forming unit that's in the spleen and colony-forming unit that's in the lymphocyte lineage. So while the pluripotential stem cells can give rise to any blood cells, these two categories are only going to give rise to a subset, not all of them. That's why we call them multipotential. Another term we use in development is totipotent or totipotential, and that would only be used for the fertilized egg because everything is going to come from that one very early stem cell. So we're going to look at the CFU-S and the CFU-Li. And as we put this sort of into a flow chart, here we can see our pluripotential stem cell at the very top, giving rise to our CFU spleen and our CFU-Li. These are going to be our multipotential stem cells that are going to give rise to our different lineages that we have here for our lymphocytes. And the CFU spleen is going to give rise to all of our other. As you can see, we have other categories of stem cells that we're going to walk through next. Now, from our multipotent cells, we're going to get into progenitor cells. These are unipotential. They have somewhat of a restricted capacity to self-renew. So these, it's hard to really call them stem cells because these cells are just going to be part of the developmental process. They're not really going to self-sustain themselves. We're always going to have the multipotentials producing more of these progenitors. Then we're going to have the precursors. And the precursors are totally incapable of self-renewal. Once they start down the path, they're going to have to end up being that full-grown blood cell. Otherwise, those cells will die. So we talked about hematopoiesis. I know in physiology, we certainly talked about erythropoietin, but there are many other factors that help regulate this. Interleukins, for instance, they're going to stimulate the proliferation of our pluri and our multipotential stem cells. Colony stimulating factor, this is going to impact unipotential cells of the granulocyte and the monocyte lineage to begin to clone themselves to produce more of their destined cell. We've got thrombopoietin, stimulates the development of platelets. And steal factor, this is going to act on most of the stem cells that we have in the bone marrow, and including bone marrow stromal cells, those sort of connective structural cells in the bone marrow. It's going to cause them to proliferate at an increased rate. So first, we're going to look at erythropoiesis, and we are going to produce red blood cells from our CFUS, that is our multipotential stem cell. And with erythropoiesis, we are going to see that we first form a burst-forming unit, erythrocyte, and then we form a colony-forming unit, erythrocyte. So these are our progenitors and our precursor cells. So CFUS cells, these multipotential cells from the spleen, respond to erythropoietin. Erythropoietin is produced in the kidney. It's going to cause these cells to divide and form first through our progenitor cell, then our precursor, the CFUE cells. And the CFUE cells that are incapable of self-renewal, they're going to form proerythroblast, and this is the first precursor that we really recognize and can actually identify. Now, in lab, proerythroblast, we're not going to identify. That's not going to be what we're going to do. We know this is the first precursor, but not the first one you're going to be responsible for identifying in lab. Now, as red blood cells are going to progress now through these different precursor stages, what's going to begin to happen is that we're going to decrease gradually in size, nuclear size, cellular size. We're going to increase the amount of hemoglobin that's present, so we're going to go from a blue color in the proerythroblast to the nice pinkish red color that we know in our erythrocytes. And we're also going to condense the DNA, condense our chromatin more and more and more until we eject the nucleus. Now, this is a rather ancient picture from a book, but it does a really nice job showing you these progressive changes. Here we see essentially no hemoglobin down to full hemoglobin. Here we are looking at nuclear size, and we are showing how that nucleus becomes more and more compacted. Okay, this is also showing the size of the overall cell decreasing. Here we can see the DNA and the chromatin fairly dispersed, but that chromatin is going to become much, much more dense as it's compact tightly down into just a pinpoint pupil, very, very black spot. And then when you put all this together into actual illustrations of what the cells look like, here we can see the proerythroblast all the way down to our mature erythrocyte. So we said that proerythroblasts, we're not going to identify that. The first real stage we're really going to look at is the basophilic erythroblast. This is the first one we're going to see in our histological slides. In this one, we're going to have a relatively large cell. It's bluish in nature, and the chromatin is going to begin to condense. And we're going to see it almost as a mottled look for the chromatin. Now, the third step, the polychromatophilic erythroblast, this is where the chromatin is really going to start to look like strands of genetic material, much more than anything we've seen in a nucleus before. The orthochromatophilic, this one is very easy because this one is the jet black, solid looking nucleus in the very center. Sometimes it might be slightly offset, but it's that dense black spot that's going to help us understand that we're looking at an orthochromatophilic erythroblast. It's also at this point that we're going to start to see really that change from the blue to the more reddish color. Now, the reticulocyte, this is going to be a red blood cell that is still in the bone marrow. It's not necessarily taken on its biconcave shape, and it hasn't completely lost the bluish color to the cytoplasm. So it hasn't completely filled with hemoglobin yet. That's how we'll identify that. And then when you get out into the circulation, peripheral blood, that's where we'll see our mature erythrocyte, nice red filled with hemoglobin. There's our illustration showing our basophilic erythroblast, polychromatophilic, orthochromatophilic, reticulocyte, and then our mature erythrocyte. Here's just lineages, and again, we're focusing right now on this left-hand boundary here, basophilic erythroblast. Then we're going to have our polychromatophilic erythroblast. I know they don't have this. This is an older view. Here we still have the nucleus, and so it's referred to as a normoblast here. This is our orthochromatophilic erythroblast. There we have our reticulocyte, and finally our mature erythrocyte. Now for our granulocytes, we're still going to derive these from our colony-forming unit spleen, and that's going to be for our eosinophil and our basophil. However, the neutrophils are going to be from a different lineage. These are going to come from our colony-forming unit granulocyte macrophage lineage. So the CFU-GM, this is also coming from the CFU spleen. So this is going to be our multipotential stem cell that's going to give rise to a neutrophil and a monocyte macrophage lineage. So we can see we have different multipotentials happening here. Now in our granulocyte lineage, whether we're coming from the spleen or we're coming from that granulocyte monocyte multipotential stem cell, we're still going to go through these specific steps. The myeloblast, that's going to be very early, just like our proerythroblast, and basically they're going to look fairly similar. But then we get into the promyelocyte, and the promyelocyte is going to be the first one we're going to have any chance of really recognizing and distinguishing. It's going to be much larger than we see with our proerythroblast. But where we're specifically going to be able to distinguish in lab the different types of granulocytes we see as our stage 3 is going to be the myelocyte. And in this one we can begin to see the specific granules, so we can call this a neutrophilic myelocyte, a basophilic myelocyte, and eosinophilic myelocyte. And when we see those granules first, that's when we call it a myelocyte. Now the next step, again, as the cells continue to accumulate those specific granules and the nucleus really begins to change shape, this fourth step, the metamyelocyte, is when we begin to see a really indented and kidney-shaped nucleus. We'll have the specific granules, and we'll term them as such, but now we're going to call them a neutrophilic metamyelocyte, and so on. Now, when we finally arrive at this extreme of this horseshoe-shaped nucleus, really thinned out boundary connecting our lobes, then we call this a stab cell or a band cell, again preceding that with either the eosinophil, basophil, or a neutrophilic label. And then finally, once we get out into the circulation, we've got the specific granulocyte, and we can just name those eosinophil, neutrophil, or basophil. Again, here we can see our lineages. We've got our myeloblast, promyeloblast, giving rise to our different stages that we see here. Myelocytes, metamyelocytes. We've got our band or stab cell, nice horseshoe shaped. And here we have our mature granulocytes. Here's an illustration somewhat comparing the erythrocytic pathway with the myelocytic pathway. And again, here we're not going to really compare our proerythroblast with our myeloblast. You can see basically same size, very difficult to see. But the promyelocyte's really huge, and you can begin to see some granules, but nothing specific. Once we get to the myelocyte, that's where we can see our specific granules. Then the metamyelocyte, we can see we start to indent the nucleus. Our band or stab cell is very much elongated, horseshoe shaped. And then finally we have our mature granulocyte. Now, monocytopoiesis, this is the lineage for our monocyte macrophage family. We have our colony-forming unit spleen. This is going to be one of our multipotential stem cells. We have granulocyte monocyte lineage, and that's going to split into just the monoblast lineage. So here we have these precursors, these progenitor cells, before we get into the promonocyte and the monocyte. And again, these we're not going to be identifying in the marrow. We're going to be looking for the monocyte versus the macrophage in tissues and in our peripheral blood. Platelets, they're going to come from the colony-forming unit spleen, and then we're going to have colony-forming unit MEG. And this MEG's precursor is going to be the megakaryoblast. These are going to be very large cells, and they're going to undergo a really unique process. It's called endomitosis. These cells are going to begin to divide, but the nucleus is dividing without any cytokinesis. So you're going to end up with cells that have multiple nuclei, and they may actually be polyploid up to 64N. We're used to 2N, but certainly not 64N. Now, once we have gone through that process, now we're going to have a megakaryocyte. And these can be upwards of 100 microns. These are going to be the largest things you're going to see in the bone marrow. And these begin to pinch off these small portions of their cytoplasm in these little membrane-bound packages, and they're going to just release these into the sinusoids of the marrow and go into that hematopoietic compartment and enter the circulation. And these are going to form our platelets. And once we've removed most of our cytoplasm, then what's left of our nuclear material, it simply degenerates, and macrophages that are resident in the bone marrow, they remove that material by phagocytosis. Lastly, lymphopoiesis. The colony-forming unit Li is going to give rise to our progenitors, CFU-B and CFU-Li-T. The CFU-BU, this is going to give rise to our B cells, and the CFU-Li-T is going to give rise to our immunocompetent T cells. These are going to travel to the thymus where they complete their development. They're screened to see if any of these cells recognize self-markers, in which case if they do, then they're going to be destined for apoptosis and destruction and never leave the thymus. Remember, only about 20% of the T cells that enter the thymus ever leave and function in the peripheral body.